But now we went to we're a well-organized patient advocacy group, and we spread awareness to the world about DUP15Q syndrome. We have 7 countries represented here this week, which is amazing that we have people from, we represent, I think, 74 countries. I think it was the last count because we did the database poll. It's amazing. Spreading awareness to the world about DUP15Q and upholding our vision of advocacy and awareness. This did not happen by accident. It took the involvement of some remarkable people, many of whom are in this room today, and still engaged and still involved. And by their work and dedication, they brought us to this point. Under the guidance of these incredible people, the DUP15Q Alliance has attracted leading clinicians and researchers to help figure out the puzzle that is DUP15Q syndrome. And through grassroots fundraising by our community, DUP15Q Alliance has been able to play a leading role in pushing the field of DUP15Q research forward. Together we have believed, and together we will achieve. This morning we're going to focus on that the point of finding targeted therapies and providing medical care. So, we want to answer the big questions, the questions that I'm asked almost every time I have a new family come in. Will my child walk? Will my child talk? What's the difference between IDIC and interstitial? How many cases are there in the world? Will my child have seizures? Will my child have autism? Will they wake up tomorrow morning? And how long will they live? There's some general clinical information out there that can be used to make assumptions about these things, but in order to answer them with certainty, there are specific types of research we need to start doing, and one of them is called the natural history study. I know that Shefali mentioned it yesterday. Does anybody in the room know what a natural history study is? Nobody raised their hand, just so you know. So I'm going to talk to you about what a natural history study is and why it's important and why we're going to be doing one. So, natural history of a disease. The natural course of a disease from the time immediately prior to its inception, progressing through its pre-symptomatic phase, different clinical stages, to the point where it has ended and the patient is either cured, chronically disabled, or dead without external intervention. That is a morbid way of saying, what does it look like over time? And so how do we do that? How do we figure out what it is? The importance of natural history studies and natural history course in rare diseases is a little bit different than the normal course of a lot of disorders. Diseases like DUP15Q syndrome or disorders, I hate when they call it diseases, but we're considered a rare disease. So when you hear it, it's just because that's the umbrella we fall under. So they're usually poorly or incompletely understood, and generally the low prevalence leads us to have less understanding about them. We're small populations, and there's limited opportunity to study the kids and also to replicate the studies that are done. When you participate in research like that's going on here at UCLA and Geisinger, we bring these rare populations together, so it's really important to engage when you have the opportunity. So there's usually little precedent for drug development within these rare disorders. So drug companies aren't really interested in rare disorders, and they haven't been in the past because they're small groups. They don't have the opportunity to make money, and that's really sad. But when you get people together like patient advocacy groups like DUP15Q syndrome, where you can represent the entire world's population, people start noticing. And when you get as well organized as DUP15Q syndrome or DUP15Q Alliance has, people start really noticing. So we need a solid scientific base upon which to build this research program and to get pharma and other interventions really interested. So we're going to do a natural history study, and we're going to track the course of DUP15Q over time. A natural history study is a very, very expensive thing to do. However, there are people who are interested and will be funding it. I can't say names, but it's going to happen. So, what we're going to do and what our goals are and what Sean's going to kind of tell you how we're going to do it technically, is we're going to track the course of DUP15Q. We're going to identify the demographic, genetic, and environmental and other variables that correlate with DUP15Q syndrome and the outcomes in absence of treatment. This is not absence of treatment meaning that you don't go to the doctor and you don't get your seizure meds. This is absence of novel treatments, so novel drugs or novel things that we can try out. Many potential uses is we can change patient care. We can make your patient care better. And in these cases, we're also looking at pharma partnerships. So we're going to inform drug development on this one. We're going to create a knowledge of DUP15Q that's essential for rare disease drug development and moving on to better clinical care. So the key points of this: natural history study data contributes to the scientific foundation upon which treatment interventions and drug development programs can be built. We want the rational, scientifically based drug development requires the understanding of the disease. Natural history describes the disease before the introduction of investigational drugs. And they're most informative when natural history studies data is available early in development, meaning right after you get your diagnosis, we want to know what happened up to that point. So what caused you to get your genetic testing, what happened afterwards, and how that kind of progresses. And patient and caregiver involvement is really important. We want to engage all your stakeholders in this. This means parents, this means researchers, this means your teachers, the people who interact with your kids on a daily basis, and also your clinicians as well. We want them to be actively involved in this data collection. So how do we get this data? So most of you know that we have a registry. When you came into the Alliance, you signed up and you probably spent 2 hours, if you actually completed it, you spent 2 hours in front of a computer and maybe answered some questions about seizure history, autism, seizure meds that you're on in general. The problem with the registry is it takes 2 hours to complete it. It was too long, people didn't do it. Our registry only represents about a quarter of our patients that we follow. So what we're gonna do is we're gonna change that up a little bit. We're gonna use the technology that's available to make it more friendly to you. We're still gonna do an intake, which will take a little bit longer, but it's not gonna be 2 hours. It's not gonna be, you don't have to find everything at the same time. But then after that intake's done, we're gonna email you, or the system's gonna email you every 3 to 6 months and say, hey, has anything changed? Has your kid been on any new meds? Have they changed any new therapies? Have they started having seizures? Have they stopped having seizures? And if you say no to all these things, it'll take you 30 seconds. If you say yes to some of them, it might take you more like 5 minutes, maybe a little bit longer. But the purpose of that is the system is gonna remind you, we're gonna remind you to give us a data point. We're interested in what has happened over the last 3 to 6 months. If something really big has happened, we'll probably call you and give you some follow-up questions, but we really wanna know at regular intervals what's happening in the life of your duper. We wanna know anything that could possibly inform what the progression of this disorder is and what can answer these questions, these big questions that almost every parent asks. So we set up our study with a very specific purpose, which is intended to be comprehensive and granular, meaning that it's going to ask big questions and it's going to follow up with littler questions if you answer the big questions in a certain way. And it's intended to describe DUP15Q from the bottom up and also from the top down. Some things you'll wonder why we're asking that. And we have thought about each and every question that goes into this. We want every data point to be used. We want every data point to be interesting to researchers, to clinicians, and to industry. So if you ever have a question, if you don't want to answer anything, feel free to call me. I'll just tell you why we're asking that question. Or if you don't want to answer it, that's fine. You can just say there's not applicable. Okay, so the importance of being part of this natural history study is, so when you get that email that says, would you like to be part of this, and clicking on yes, when clinical trials come up, when we have drugs that can target different symptoms in DUP15Q, this is your best way to get in there. We know what your duper looks like, we know what their symptoms are, and we know if that drug can target that symptom, you're gonna be a good candidate. Also, we know you're a good participant. We know you're compliant, meaning that you read and fill out all the forms that we send you. We know that you're gonna follow up with us, and we know you're gonna answer the questions when we ask them regarding that intervention. So participate early, participate often, and participate to the end. So we have somebody from the Autism Brain Net here today, and she's gonna talk a little bit about postmortem things. The end is also important. So we don't like talking about it, and I don't want to talk about it for long because I cry every time. But there's a possibility that dupers do pass away unexpectedly. When that happens, it's still important to participate. Alicia will tell you a little bit more about what that means in terms of tissue donorship when it comes to DUP15Q and what that whole process looks like. But that's kind of the end point. When we have all of this information up to that point, so from diagnosis or sometimes birth, all the way through the end of life, that's really what's going to inform us the most. Also, if you are willing to be a tissue donor, that information that you've given in the past can connect up with it, and we can really say, well, this is what it looked like, this is what happened at the end, what happened leading up to that? What can we see that maybe can correlate with that end result? To possibly give us more information about why there are passings the way they are, okay? Okay, now, being a good consumer of research, clinical trials, and natural history study, not everybody's going to qualify. And I just want to put it out there that there is a reason why people don't qualify for natural history, or not natural history, for clinical trials. It's nothing personal. It is not something to get upset about. You don't need to call me and yell at me to say, why can't I get that drug? The targets of different drugs that we're thinking about have specific mechanistic things that we want to look at, meaning that it's saying like this, it's targeting this symptom, it's targeting this thing. So maybe your child doesn't have that. That's not one of the symptoms of DUP15Q that your child has. So when it comes up to that, know that we're looking at other ones as well. So if your child doesn't have that symptom and that drug is not right for your child, we're looking at other interventions that might be related to your child's symptomology. So participating in research, how do we get involved? We're going to design these studies in a way that all of our membership is able to participate, even if you're not able to come to a clinic to be seen. So those questionnaires that come up through your email, they are standardized. We can score them. They're going to give us very valid clinical and just descriptive data about your duper. And then, one thing about participating in research, and especially when you talk about clinical trials and that sort of thing, I want to talk about informed consent. Does anybody know what informed consent is? Other than Paul Karch, because he's a lawyer. So informed consent is something that you're going to go through every time you go into a research study. It's that long thing at the beginning of everything you say, click accept. Informed consent is also like Apple iTunes, when they say accept, 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 it's 17 million pages. That's informed consent. It's something that you read, most people gloss over and they don't read the details. When it comes to doing trials, whether it be behavioral, whether it be drug trials, or whether it be kind of any research, make sure you're reading the fine print. These are things that I know we're grasping for answers, we're grasping for anything that can help our dupers, but when it comes down to it, as a parent, you are the best advocate and you're the best carer of your child. You want to make sure that you're reading through the fine print. You're wanting to read the adverse events. You're wanting to ask questions. Don't just take the word for it that this is going to work, this is going to be the best, this is going to be amazing. You want to ask the tough questions because there are going to be adverse events listed under there. And are you prepared to deal with those if they come along? It's something that I know that we really want to get involved in drug trials, which is great, but they all come along with their own side effects. I know that a lot of you guys who are on epilepsy meds, you know what side effects mean. There's a whole talk about balancing side effects versus seizure control. It's the same sort of thing as you're introducing new drugs as well. So when you ask those questions, make sure you're educated when you're going in there saying, I'm ready to do this. Also, when you are enrolled in a drug trial, or when you're enrolled in a clinical trial of any sort, whether it be behavioral or drugs, making sure that you're adhering to study participation guidelines. That means a big thing that's been happening in general clinical trials is social media is a confounding variable, meaning that you find somebody else who's in the trial, and then you talk about it on social media, and that's not good. We want to know if the trial is actually working. We want to know if the treatment's actually working. So if you enroll, you may say, we may be on the drug, or we may be on this treatment, but not talking about it on social media, because what ends up happening is it becomes a subjective versus objective. Subjective means, I feel this is happening, I feel like she's getting better, I feel like this is, like her seizures are decreasing. But it's not objectively saying, I know they're decreasing, I know we're counting, I know the count is becoming less. So making sure that you're not being swayed by anybody else who might be in that treatment. It's happened in a lot of different drug trials that are out there, that social media has become a huge issue. So as we're moving into this, because this is definitely the future for DUP15Q, I just want to put it out there and make sure to make you aware and educated about this, because that's one of the things we want to do, is make sure that you guys know going into this that we want to set it up for success. We don't want these small pitfalls that we can avoid being the disaster that could happen. So, okay, so how are we going to collect this data? And so Sean is here from the University of Tennessee, your lab owner. Right, is that what I say? Okay, so she's going to talk to you about the database that we're setting up to collect the natural history study data. You going to put it on? Do you want to introduce her? Oh, yeah.
Is this also for outside America? Is this for the whole world? Do we have
Yeah.
a chance to be in it?
Yeah, so you do. No, you do. So that's the other thing. So the DUP15Q Alliance will never be, I said this in the international meeting, but I'll say it here. The DUP15Q Alliance will never be DUP15Q Alliance America. We will never be DUP15Q Alliance United States. We will always be DUP15Q Alliance. We represent every patient that comes to us or finds us throughout the world. We support any other organizations that want to create a country-specific organization, because we know that there's different care, there's different systems, there's different recommendations in each country, and they really do want to incorporate. But regardless, we have the best chance of actually finding answers if we all work together, and that means worldwide. So when the natural history study comes out, it is inclusive of all international patients. When it comes to the clinic side, because there is going to be clinic visits involved for some people, as long as you can get to one of the clinic sites, and they will be in the United States to start with, there's a possibility that they can extend. It's something that we're discussing. But it is available to everybody internationally. If you're a registered DUP15Q Alliance member and we've verified that you have your genetic diagnosis, you will get an email saying, we're starting the natural history study, you're ready to be in the database, here you go. So everybody will be in there. And so when we get requests for information about you guys, meaning just the demographics, we don't give specifics, but we give them that information. We say we have 4 families in the Netherlands, or we have 5 families in China, and they are interested in this as well, because obviously when you're talking about industry, they're interested to see where they can put things to. They're interested to see what their demographic that they can eventually release the drug to.
Okay, so I'm Shauna Ajayi. I work for Lebanon Children's Hospital, and we are the data management core for the research database, and that's what I'm going to cover today. Okay, so just an overview, I'm going to go over the background and objective of the database, also the purpose, where the data is going to be stored, why it's important, and how easy it is to use, who can access it, who to contact if you're interested, and that's about it. Okay, so the database was established in 2014 with a collaboration with Dr. Ryder at the University of Tennessee Health Science Center and the DUP15Q Alliance and other clinics, clinicians, and researchers. And so the purpose of the research database is to establish a national de-identified database that can be used for research purposes. And so what we did is create a standardized collection of variables that will be captured across all clinics who are participating. And they're a combination of manually entered data and data completed by the families. So each clinic has the same standardized variables that they'll collect. And then they have the option to also create specific clinic variables that they'll collect for just themselves. If the alliance and the consortium think it's something that is useful, then they'll push that out to all the clinics. And so these are the research database measures we're collecting right now. The first one, the pre-clinic visit surveys, are completed by the families, typically prior to them coming to the clinic for evaluation. Sometimes that depends on what kind of access you have to the internet, because these are online surveys. And so during the clinic visit, you also fill out the SCQ and CBCL, and then there are online testing forms that are typically completed by clinicians. That data is typically entered either online by the clinician, or they'll have to enter it manually after they've collected the data. And so the database is set up to collect longitudinal data over time. A lot of these measures will be collected more than one time, so keep that in mind. And that's really just to show the kind of growth you make over time. And so in this bottom, other instruments are other measures that are collected. These are not standard across all clinics. Some clinics do some of them, share some measures, some just do all of them. It just depends. So these are the measures that they decided that they wanted to collect additionally, possibly what they already do in their clinic, and they just want to have it in a centralized place. And so right now, the data is stored in REDCap. So REDCap stands for Research Electronic Data Capture. It is a secure web-based clinical database management system. And we support that at Lebanon Children's Hospital within our biomedical informatics group. And so what we're trying to do, right now the database is set up that each clinic has their own database. And so each clinic stores identifiable data that is only accessible for people who are clinicians and researchers within that clinic. So nobody outside of that clinic can access that data. And so to combat that, we're transitioning to REDCap Cloud. So REDCap Cloud is just another form of REDCap, but it's cloud-based, and it's just a clinical research data management system. It is secure and HIPAA compliant, and 21 CFRI 11 compliant. So what it's going to do is combine all of our databases into one database. So the membership database, the registry, and the research database will all be housed within one site. And so we don't have the trouble of trying to get other clinics to access your data. So if you go to one clinic, and you go to another clinic, they can all access the data at the same time. So we're housing it within a centralized place, and that REDCap Cloud supports online data collection. And so the purpose of this is, of course, to collect data on patients with DUP15Q syndrome. So we want to collect your data, and we want to use it to investigate further into the syndrome, and possibly develop new treatments, new research projects, and also dive into clinical trials. So having that information on hand lets us know who is eligible for those clinical trials if they come up, or having that data will allow us to further investigate and develop new projects. And so why is it important for you to participate? First, it allows you to contribute to the knowledge that we have. So you are our best knowledge source because you live with it every day. And so us capturing that data from you is important so we can better understand your needs moving forward. Also, it improves and encourages the development of new treatments and new research studies. And so with that data, we want to be able to take it, analyze the data, and see what we should do moving forward. And so one example I have is our gastrointestinal study that we did have at Le Bonheur. And that was generated by a lot of our patients were having GI problems. And so they didn't have, there was no research on a prospective GI studies in this population, so that's what we did. So the data that you provide to us allows us to further generate new studies, new hypotheses, and new treatments that can help you. And so another reason that you should participate is that you are helping other families, not only yourself, but others. So of course, for a research project to work, we need more than one patient. And because this is such a small population, we need to pull all of you together to develop these research projects. And it's hard for clinicians and researchers to see trends with just one person. So we want to be able to capture all of your data together and see what it looks like and then move forward from there. So not only are you helping yourself, but you're helping the other families. And of course, Dr. Shefali mentioned it yesterday that a lot of these measures are expensive and may not be covered by insurance and things like that. And so everything that you do or everything that is done may not be done in a typical clinical experience when you come to clinic. So not participating really limits your ability to have access to those treatments, new testing measures, new medications if you're not participating. So we want you to be able to assess these new treatments, assess if these tests are working, but you can't really do that if you don't participate. And so typically we're trying to make it as easy for you to use as possible, and this is just it's kind of hard to see, but this is what REDCap Cloud's interface looks like. This is just an online form. So all the forms will be accessible online, so you can access it from your phone, from your computer, and they'll just be quick surveys typically, or questionnaires that you'll fill out online and submit, and they'll save to your child's record. And so many of the surveys will be sent via email, and that's hard to see too, but this is just a sample email, what it may look like. It'll give you a link to click on. You just click on it and open up the link. It'll tell you what the survey is about. If you have any questions, you can always email back or contact Vanessa or the clinician or coordinator, whoever sent it to you, whoever you're seeing, and then so you can fill it out. So it's all electronic, and so with REDCap Cloud, again, everything is stored in a centralized place, and that's the biggest thing. So that membership database is connected. All that data is connected. Right now, they're all housed in separate places. It's very hard to combine that into one database because we all have different identifiers in each database. Maybe they're not consistent across all of them. And so REDCap Cloud is solving that issue that we're having. REDCap Cloud will also send you reminders to complete surveys. If you ever forget, they'll send another email reminding you to do that, so you never have to remember that. And we're trying to make designs to make it as simple as possible. So like Vanessa said, we're going to do short surveys that shouldn't take too long over periods of time. And again, it links all the databases together.
And you don't have to remember a password or login.
Right.
They'll send you the email and you don't have to log in. Like, everybody does email, I
Yes, so you don't have to remember login as long as your email stays valid. And of course, you can always update it. As long as it stays valid, you'll get those emails. And make sure you check your junk files that didn't go there. Yeah. Sorry. And so who has access to the database? Currently, clinicians and coordinators at DUP15Q have access to the database. Okay, I see the difference. So clinicians and coordinators at the clinics have access to the database, and of course the DUP15Q Alliance owns the data. We currently have about 4 participating clinics, and Memphis used to be a participating clinic, but they have closed. And so having access to this data, of course, limits the need to collect data points over and over again. So you don't need to capture demographics more than one time. That shouldn't change in most cases. So things like that, we want to minimize that duplication of data collection where it's not needed. And so in the future, we're going to intend that researchers outside of the Alliance are going to be able to have access to that data, and the Alliance will come up with policy and procedures for that, so they can access the data and formulate their own research projects with that data, even if they're not a part of the clinics seeing patients. Okay, so to find out more, you can contact Vanessa. If you have questions about REDCap and the database, you can ask me, but I'm not a doctor. And you can either contact the coordinators or clinicians or researchers at the DUP15Q clinics. Of course, some of them aren't participating, but if you think that you're interested in participating, you want them to participate, just have them contact Vanessa and we can get that set up. Okay. So just an acknowledgement, we'd like to thank the DUP15Q Alliance, Dr. Ryder, of course, for bringing my team into the project, the biomedical informatics core of my team at Le Bonheur Children's Hospital, all the participating clinics, and of course you, the families, because without you, we wouldn't have any data. All right. Any questions? No? Okay, thanks.
Oh, that's right. So all of the data that you filled out before in the registry, so if you've done the registry or you've done any of the forms, it actually is going to be uploaded. So we're not going to make you do that again. So which is good, right? Because you spent 2 hours doing it or more. So you're not going to have to do that again. The only thing is we might have you verify it just to make sure that it still is the same. A lot of times we verify that maybe a genetic diagnosis has changed. We have had families that they thought they were IDIC, now they're interstitial, or vice versa. So we'll just verify that when that data is in there, that it's correct for you, including your address and your phone number as well. That is always going to be the first question on every survey will be, has your phone number changed? Has your email address changed? Just so that we make sure that we're in contact with you guys and able to get the information to you as well. Yes. So everybody that's in the registry is going to get the consent form to go into this. So you're gonna get a consent form saying, I would like to participate in this new version.
Vanessa, in a perfect world, if you had 100% response, how many families do you think you'd actually hear from that responded? What's your number roughly of how many?
How many do I want in a perfect world?
All of them.
Well, perfect world, so 100%, but how many people have this, and how, and are part of the registry?
I would say we have 1,360 members of the Alliance right now. In terms of what we actually think is out there, it's more. There's definitely a language barrier. We learned yesterday that in China you can't search for DUP15Q, since Google is denied there. So there's definitely, I think, a lot more representation internationally. I think there's a large percentage of people that just aren't tested for DUP15Q. So they walk around with an epilepsy diagnosis or an autism diagnosis and a microarray is never done. The neurology and the developmental pediatric field is really moving forward in that way, that when kids, especially young kids now, that when they have issues like that and like the hypotonia when they're babies, they're more likely to pull a microarray now. One of the big things for us is we do advocate out in the clinic areas and the genetics areas to say, you need to be pulling microarrays more often. Because rather than just kind of doing all these behavioral assays, if you can pull a microarray and figure out if this kid has a genetic disorder that is known, you at least have some way to go. So we will know a little bit more about prevalence we're participating, or we're supporting the Australian blood spot screening. This is actually a really exciting project that the Alliance has actually put a financial stake in. They are screening, it's a population-based screening, meaning that all babies born in 4 hospitals around the metropolitan areas in Australia are going to be screened for DUP15Q. So meaning it's anywhere between 500,000 to 1 million babies. So what that's going to tell us is the exact prevalence of per birth, per 10,000 births, how many kids with DUP15Q do you see in Australia? There might be slight country differences, but we're going to know pretty definitively what the number is. So then we can make a better estimate worldwide.
Okay, but when you say 13, 16, that's the actual number, that's actually the number of people that have DUP.
That's the number of people that have found us. The estimates are much higher.
Okay, and how many of those are U.S.?
890 something, if I remember right. So the other thing is that when this is all running, so when I came on about a year ago, I'm doing a lot of data cleaning. I'm doing a lot of cleaning with the registry, I'm doing a lot of cleaning with the membership, and to be able to answer the questions much more clearly and succinctly, when we move to this, it'll be pretty easy to see exactly where everybody is. So we'll know, if you want to know IDIC versus interstitial on that day, all you have to, if somebody puts it on Facebook, I'll be able to pull it up and just send it right away. It'll represent everybody in our membership rather than just the 1 quarter that's in the registry right now. So when you pull that map up, or when you pull the IDICs versus interstitials or Seizures versus not, it's a very small representative non-representative population. So. I'll let you know. So if you'll get an email. You'll get an email saying that you filled this out, and would you like to be moved over to the other one?
And if we haven't filled it out yet?
You won't get an email.
Okay.
But I'll know. And then I'll email you separately. You're going to get an automatic email if you filled it out, and if you haven't, I'll email you separately and say, do you want to do this? And then here's the link to the consent.
And when?
We're estimating rolling out in late fall.
Okay.
We want to make sure, like I said, natural history studies need to be very, very well thought out with the end goal in mind. And to do that, we need to go through each variable very, very carefully. And we don't want to overextend. We don't want you sitting in front of your computer for hours and hours and hours. The good part about this as well is if you're seen in a clinic that does an ADOS with you, that's not a DUP15Q clinic, you can upload it. You can put the information in there. So we can track that as well. You can upload your genetic report, and that's a place to repository it. We are also, you'll hear about it in a little bit, we are also looking into having a place to repository your EEG. The reason being is you'll hear about it at about 10:30 as to why, but it's another place that we're interested in that kind of information as well.
I remember filling out the registry,
Mm-hmm.
but when we filled out, I didn't have so much knowledge. So a lot of the questions that were just like
Yeah.
Chinese for me. And then once we get into the thing, I always say, like every 2 months, say, oh, we should load up all the things that we didn't have when we filled out, and finally we never come around.
This will give you a reminder and an option to do that. Like it'll give you an opportunity to say, oh, I should do that. And also you can always go back and you click on the link and you can upload it again.
There's information on the current registry that we as members can look at,
Mm-hmm.
look at how many other kids have sleep disorders, how many kids are having epilepsy, et cetera. Will that information be available on the new database for us?
So the interactive part of it is a little bit different than the other one. That is, you have access to it, yes. The way right now you'd have to have access to it, so if you post something or email me, I will post it. What we're gonna try to do is on the website, post statistics, or not statistics, just descriptive statistics, meaning that how many of each, every month that we update. So that you'll see the kind of how the alliance is growing, you'll see how many numbers are going. I'm also gonna use a little bit of guilt, good old-fashioned guilt, at how many people haven't filled out their stuff, so that, that we're giving you some really good guilt. It will be available, but just in a little bit different manner. When you click on it, you won't be able to see it, but we're going to update it that on that part of the website, you're going to see how is the registry going, or how's the natural history study going. Is everybody excited about this? It's really exciting. So, well, at least for us.
Will our doctors have access
So if your doctors would like to have access to the information, so are you not seen at a DUP15Q clinic? Right? So DUP15Q clinicians do. We assign you, if you're seen at a clinic, you're assigned to a clinic and they're able to see that because that's part of how it's just structured. So if you switch clinics, then we change your person to that, they're able to see it. If you would like your doctor to, we can print them a report. You can give them, you would just have to sign a release and you can have access to it. The researchers that Sean talked about having access to it, so we do have people that are interested in us, that are kind of outside. We're going to open it up in a non-identified way, so they're not going to get any identifying characteristics of any of your children or any of you, but they'll be able to do analysis on it. All analysis that's done will be vetted by the professional advisory board first, so that we know it's a valid research study and it's actually going to answer questions that we want to answer, not something that's not interesting to us, because or something that you'd be surprised so many weird requests we get. Yeah. 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 So Lebanon isn't actually no longer a clinic, so they actually don't upload data anymore, but you can. Because the issue, so a little bit of the structure of our clinics, in order to be called a DUP15Q clinic, they have to agree to it's a business agreement. They have to say, we will upload data to the website or upload to the database. We will actively participate in your clinic's working groups. We will actively participate in everything DUP15Q Alliance. It's part of their agreement. So because we lost the lead clinician on the business agreement at Lebanon, until we find a new one, we're kind of on pause with that, but you can upload any of the information that you can, and you can sign a release and say, can I have that information to put into here? Any other questions before we introduce the next speaker? So, like I had said, the natural history study, there's an end. There is, and it's very unfortunate. However, there's a way to keep participating. There's a way to make that end really matter. And so Alicia's going to tell you a little bit about this, and I'm going to let Fernando introduce who Alicia is, Dr. Halliday.
There we go. Okay, let's get started. So, as Vanessa mentioned, I'm going to talk about something called the Autism Brain Net, and this is a way for people to donate their brains after death so that research can be done on them. And I know the topic is pretty scary, it's definitely depressing, and no one wants to think about when that time will come for their children, and even more so themselves, because then they have to think about what's going to happen to their kids after they go. But this is going to be a more research-based presentation, so you can learn about the research that's going on. And we're going to make it into a little game. So whenever I say one of the words that is on your bingo sheet, just cross it off. And I have a bag full of prizes. Don't get too excited. This isn't The Price is Right. So if you see one of the words on your sheet, just cross it off. And as soon as you get bingo, just hold back, because I want to get through. And then we've got prizes. And if we don't have the size prize that you want, then I'll send one to you. Okay. So the reason the Autism Brain Net exists is because there's really a lack of understanding of the brains of people with autism, certainly the brains of people with DUP15. It's the main organ of interest. Of course, there are other issues and other organs that are interesting to study or important to study, but it became it's a rate-limiting factor in discoveries. So a few years ago, we had one brain bank, and unfortunately, it's a long story, but a freezer broke down, and a lot of the brains or the tissue were lost in that. So the idea was to say, all right, we can't just have one brain bank anymore. We need many brain banks. And then also, there are too many diverse methods of people, slicing the brain, studying the brain, and so it was coming up with different findings. So many of you who've been around for a while have heard of the Autism Tissue Program. This is now called the Autism Brain Net. Oh, yes, and if you've already registered for the Autism Tissue Program, you're in the Autism Brain Net registry, okay? So one of the things, this came out just 2 days ago, and it's made the news. It actually made the news this morning in Los Angeles. There was a big story on CBS this morning about it. This was a study that was done on actually 202 brains in total, 100 of them, and these were 202 brains of football players. 110 of them had reached the level of the NFL. In 2008, there was a special brain bank in Boston formed to study people with football, people who played football who they thought had something called chronic traumatic injury, concussive traumatic injury. So this is when you get hit on the head too many times. People who played football were reporting things like memory loss and disorientation, and so a group decided to study it. They actually found, they started this whole thing in 2008, so really only like 9 years ago. By 2013, they had already had 69 cases that were donated, and because of this, they were able to actually make standards for what this was, which incredibly helps the field. To be able to actually define it and to show that it exists is incredible. By 2016, there were 202 cases. 111 of them were from the NFL, and they found that 99% of those in the NFL had an actual diagnosis based on a biomarker for this disorder. So this really is a great story that tells you what brain tissue donation by families who make this amazing decision, completely, thoughtful, totally selfless decision can do to help other people. So it's really important to study brain tissue. It's really important that we understand the brain tissue. We can find diagnostic biomarkers like they did for CTE. It can help explain the causes. It looks at DNA. Oh, I have one of these, so I'm gonna. When I see a word, I'll stress it so you can, for DNA, and looks at the genes or the genetics. You can identify treatment and intervention targets by looking at different cell types, because every case of autism is different. And you can build model systems. So you can look at, and I think, is Jill talking next? Jill Silverman, who's amazing, is talking next, and she's going to talk about model systems for DUP15. So your brain has 100 billion neurons and 100 trillion connections, and so you would imagine this is a Herculean task to look at all this. You have 3 parts of the brain, the neurons. One is the cell body, and that's where the DNA is. And that's that orange part. And then an axon that projects out. And then there are dendrites, dendrites, that project out from there, and that's what makes connections with other brain cells. So you have 100 billion of these brain cells, but they're making 100 trillion connections in the brain. So why is this important? The brains in people with autism, the brain cells, have greater spine densities, which means these little dots, they look like little branches on a tree. They have too many of these connections. So they're making too many connections in the brain. Instead of making 100 trillion, maybe they make 120 trillion connections. And this could possibly explain hypersensitivity to noise, to other sensory modalities. It can affect the filtering of information. The good news, the reason I mention this is because you can give a drug in an animal model, and Jill won't mention this one, but she will mention DUP15 animal models, that if you give a drug called rapamycin, which I don't know if any of your kids are on it, it reverses that. So kids can be born with too many connections, and then now they've identified a compound that turns makes fewer of those connections. And those connections can either turn neurons on or be excitatory, or they can turn cells off and be inhibitory. And some of the inhibitory neurons are found in the cortex, where the brain cells are organized into these columns. In autism, they're farther apart. They're farther apart compared to somebody who doesn't have autism. So it's possible that the excitatory or inhibitory connections that go across the neurons aren't actually happening. One of the theories, or one of the main treatments of this, I don't know if you've ever heard of this, is this transmagnetic stimulation. Has anyone heard of this, or is anyone doing it? It's pretty experimental right now, but the whole neurobiological theory behind this is that it can correct this problem. And again, I don't actually know if it can correct the problem. That's the theory. So as kids age, as all of our kids age, their behaviors change, right? Like it can't all be just developmental, like, oh, well, you know, some of them, they say, well, they have more sensory behaviors, or they have some of their repetitive behaviors have decreased over time, but their sensory issues have increased over time. Some of their language is getting worse, but their non-gestural speech is getting better. Well, by looking at the brain across time, and this comes from grouping the brains that have been collected into kids, adolescents, and adults, you can see that at younger ages, the cell size, the actual size of the cell, is decreased. But then in older people, it's increased. And this could mean that there's a compensatory mechanism going on. So the brain is saying, okay, your cells are too small. We're going to do something, whatever that mechanism is, to make them bigger. But then by the time they get into adulthood, they're too big. What is this? Oh, sorry, I already went through this. I do realize we're almost ending. Okay, so it also explains when it starts. So you can look at different cells in people at any age and figure out if there's a certain cell missing or a certain cell that doesn't look right. When does that cell first emerge? When does that cell first kind of show up? And this is one that looked in the cortex and the hippocampus, HIPPOCAMPUS. And it shows that they don't develop properly, and it happens prenatally. So in autism, at least, this is pretty important because there is this continuing and lingering theory that there's something going on with mothers who don't treat their children right, and that's why autism emerges. So it's very, very important to show that autism, in fact, is something that originates prenatally. Now, there's been a number of brains, there's a total of 9 brains from the DUP15 community that have been contributed to the program. And because of this, researchers have been able to actually look at the brains of people with autism without DUP15, and then also look at the brains of people with autism with DUP15, and been able to say, all right, well, what are the differences? What are the differences that are specific to this mutation? And they actually found something interesting. They found an excessive, what's called beta amyloid plaque formation. Beta amyloid, and especially this beta amyloid, is what has been attributed to the causes of Alzheimer's disease. And so using this information, people can prepare for the lifespan. I mean, I'm not saying every person with DUP15 is going to have the same symptoms of Alzheimer's disease, and I'm not even saying that it's going to be the same period, but it's something to actually help either explain the symptoms, and then maybe potentially the field of Alzheimer's disease is going a lot faster than our field, so help with treatment targets, and then actually predicting outcome. It also helps understand the causes of autism, especially gene-environment interactions. And this is a study by Janine LaSalle, and it shows that people with DUP15 had higher levels of PCBs. Does everybody know what a PCB is? It's a polychlorinated biphenyl. And if you haven't heard of it, good. It was outlawed in the 1970s. It was a chemical that was used in manufacturing. It was found to be highly carcinogenic and toxic. And so in the 1970s, they banned it. But the problem was these PCBs don't just like disappear. They stay in the environment. So even though people stop producing them, you're still exposed to them. For whatever reason, in the brains of people with DUP15, the levels of PCBs were higher. Now, scientists are now looking into why that is and how it affects symptoms, but that was something that they could only discover with the use of brain tissue. Finally, some of the cellular changes, I'm just going to summarize this, are, I hate to say the word worse, but they actually are more profound in people with DUP15 and autism than people without DUP15 and autism. And that can, of course, explain some of the behavioral symptoms. But one of the things that's also thought to maybe contribute to is the incredibly high rate of sudden unexpected death from epilepsy in families with DUP15. So how do we do this? How is the brain net organized? I mentioned the downfalls of having one place for all the brains to be. So now there are 4 nodes. One is in California, one is in Texas, one is in Boston, and one is at the Mount Sinai School of Medicine. The other's at UT Southwestern or UTSW. And we have really tried to make people who make this decision, not just to do this at that time, but also to register, to learn more information, are superheroes. They're considering this option, not for themselves, because it's not the easy one to make, but they're superheroes to make sure, for the whole community. So we have families across the United States that have agreed to be goodwill ambassadors for the program, and their pictures are here. We also developed information with families' input. It's important. The marketing program used to focus on individuals who had died, and that's really upsetting. It's the reality, but it's really upsetting. And also it used to be all about what the process versus what it contributed to understanding autism. So we have site-specific materials, one of which has been handed out to you. If you want to just learn more, you don't have to agree to donate. You don't have to register for the program. All you have to do is learn more, and you will get a scientific newsletter. Let me skip to that. You will get a quarterly newsletter that talks about scientific findings, educational opportunities, things that are going on in the community. The next newsletter, we're going to profile this meeting, so you can learn about the science meeting that happens in the next couple days. Please fill that out. All we need is your name and your email address, and we will make sure that you get a quarterly newsletter. Again, this is not an intent. There's this kind of perception that we are going to follow you around like Big Brother. There's 4 of us, so that doesn't happen. We also work with medical examiners. So medical examiners are people that work with the family in some states at the time of death to determine cause of death. And we work with them to make sure that they are collecting not just cases of people with autism or DUP15, but cases of people who don't have autism or DUP15, because the science needs groups to compare. So that is it. I think we're running a little over, so I didn't want to talk too much. If Who got bingo, by the way? Did anybody? Oh my goodness, we have, so I have a big, I didn't bring it up here, we have hats and t-shirts, and if you didn't get a hat, if we don't have your t-shirt size, just put your t-shirt size on your card and I'll send one to you. If you got bingo, you can go ahead and come up here with your card. Thank you guys so much for listening. Again, if you have any questions, yeah, did you get bingo? Yes, and okay.
The other thing is that if anything ever happens that you need to get in touch with the brain bank. So something happens, you can always call me night or day, and I will contact contact Autism Brain Net for you, night or day. My cell phone is always on, and you can always get in contact with them if something happens through me. So we're gonna switch the order up a little bit. Dr. Silverman's having a little bit of a technical issue, so we're gonna go on to the next speaker.
Morning, everyone. So it's time to talk about research. I'm very excited about this. I get very excited about research. The reason I'm excited about research, and I was excited about these clinical talks too, because they're all part of the same thing of solving the puzzle that we need to solve in order to get to one of our core mission principles, which is targeted treatments. So I know all of you saw Vanessa's mission statement up there. The DUP15 Alliance wants to support families, we want to promote awareness, and we want to get to a point where you all have more treatment options for your children to help them live fuller, more enriched lives. So to that end, so we have great advances in terms of clinical databases. Working backwards, we want to get pharmaceutical companies to do clinical trials. For them to do that, they need to have a network of clinics, they need to have data. They also need to have drugs, and to get to drugs, they need to figure out what they're going to look for. So to solve those pieces of the puzzle, we have many clever and dedicated researchers working on many different clues, and among them are two of our DUP15 fellows who we funded a couple of years ago. The first of those, so we're going to have three talks today. One is on, it's kind of nice, we're covering all three main models. We've got stem cell models for developing insights into human cells as a model of DUP15Q. We've got animal models, we've got flies, we've got mice. All those things are represented here today. So to start off with, we've got James Fink, who's our DUP15 fellow from 2 years ago at the University of Connecticut Healthcare Centre, and he's going to talk about his work on stem cells, passing electricity through them, and what we've learned from that. Come on up, James.
Thank you, Guy. Good morning, everyone. As Guy said, my name is James Fink. I'm a fourth-year graduate student in Dr. Eric Levine's lab at the University of Connecticut Health Center. And today I'm going to be talking to you about the project that was funded by DUP15Q 2 years ago called Hyperexcitability and Stem Cell-Derived Neurons from DUP15Q Patients. So first off, what does this title mean? It's sort of very wordy, and some of those words are a little long. So first off, what does excitable mean? So I just put this picture here because I think it sort of depicts well what we mean when we talk about excitability in neurons. So these are neurons that, how easily is it for them to sort of become active? So there's some baseline activity. You could think of, things like graduation or births or, big family events are things that get people excited. Maybe ordering a pizza gets you excited, but not as excited as graduations or the births of children. But for some people, those small things might get them equally as excited as some big things for other people, right? So there's different thresholds for what we call excitability in people, and this is also true for brain cells. So the thought behind this is, and this is true in the field as well, so not just for DUP15Q, but that hyperexcitability, so people or cells that are too excitable, potentially can lead to seizures. And really, the brain's way of communicating with each other is through electrical communication. So they send electrical signals to each other. And you can think of too much electrical activity being sort of this overwhelming amount of activity that then can lead to seizures. And so I think of sort of this picture here I got from a Chuck E. Cheese picture about like bunch of little kids playing around, lots of excitement, lots of energy, and can be a little bit overwhelming. So that's hyperexcitability. Next is neurons. So neurons are brain cells, and this was just talked about in the last talk. And so there's lots of different types of them. They have lots of different shapes. And then, again, obviously there's billions of these in the brain, and they're microscopic. Let me see if I can. And then the other thing is patient-specific stem cells, which Guy also mentioned when he introduced this talk. And it was thought a long time ago that, let's say someone who goes to college to do engineering, then they become an engineer. And the way it worked in science was that once you became an engineer, you could not go back to school, learn something new, and then change your career, right? So it's thought that once a cell was a neuron, it stayed a neuron, and there's no way it could ever revert back to a stem cell and become something else. But in about 2006 and 2007, it was discovered you could take other cell types, so in this case skin or blood cells, and then turn those back to stem cells, and then go from stem cells to neurons. And this is really an advantage because before that time, the only way you could get brain cells is through what we've been talking about, postmortem tissue, or fetal tissue, or fetal stem cells, which all have ethical issues surrounding them, and are not the best case in terms of collecting large samples of cells. In this case, anyone could get a skin biopsy or get some blood collected, and then have a huge bank, and then use those to go to stem cells, and then stem cells to neurons. So this is a way to study lots and lots of samples without having to sort of use compromised tissue or tissue that's been frozen and so on. So we have lots of neurons, and for this talk, we're going to have non-patients in blue, and there's many different, we have about 10 different non-patients here. We have, in this talk, 4 different DUP15Q patients, which include 3 of the IDICs, but also an interstitial triplication. So in all cases, these patients have 3 copies of this region. I know there are also interstitial duplications, which we have, but we have not yet studied, but that's sort of the next step. We have actually an individual with a paternal duplication, and then we also have some Angelman syndrome patients, since this is related to DUP15Q. And it's also a nice comparison because there are some overlapping symptoms, but there are obviously very distinct disorders, and so we want to sort of compare what does the actual cellular stuff look like. So again, I want to remind you that neurons are brain cells, and so how our lab studies them is we put these brain cells onto a glass plate, put them underneath a microscope, and approach them with this very, very, very sharp microscopic glass pipette that's attached to an electrode that sort of picks up electrical signals from neurons. And so this is the glass pipette here in the picture. Here's the neuron. And you apply a mild suction, and you can actually pop into the membrane of the cell, and this allows you to listen in onto conversations between neurons. So we're really going to be listening to sort of the chatter, I'll call it, between neurons. So if we break these up into the conversation, there's one part of the conversation and also the other part. And these are represented by neuron A in green and neuron B in blue. And they talk to each other at connections called synapses, where one neuron A releases chemical into neuron B, and that sort of results in the conversation. So we actually have neurons that look like this in our cultures, and so these are derived from our patients and non-patients. So here's a green-looking cell. Here's also one that looks like the blue I drew. And there's 2 types of communication. So one we call synaptic activity, which I'll say is all talk, sort of cheap chatter. This is Terrell Owens here. He's actually quite good at football, but he has done a lot of talking. He's pretty famous for excessive talk and not as much support in his play. And then we also have something we'll call taking action. And this is called an action potential. And this is actually going out and doing something. So we'll put Tom Brady here since I'm not a Patriots fan, even though I am from Connecticut, but we'll sort of counter him here to TO. So we look back to the synapses now. We can see this is what an action potential looks like. It's this large electrical signal. So I'll put Tom Brady there. And then the synaptic activity, the TO, are these smaller blips that can change in sort of their volume, let's call it, right? So there's some small volume events and there's large volume events. And the closer you get to this dotted line that we call threshold, the more you'll fire an action potential, which is this sort of activity, taking action. Now, I mentioned this earlier, you have a conversation, you could be talking about anything, and in some people, if you talk about something like ordering pizza, they can get super excited, but for the most part, this really large excitement will come from much bigger events, right? So graduations and weddings and things like that. So here's some actual data. This is an issue with using PC to Mac, but if you remember the colors I showed you earlier with the little people I drew, Controls are in blue, dupes are in purple, the paternal duplications in green, and Angelman's are in red. These are 9 different non-patients, 4 different DUP15Q patients, 1 paternal dupe, and 3 Angelman subjects. And this is looking at the number of those action potentials. How much action do cells from these controls versus dupes versus paternal dupe versus Angelman have? And you can see that across all the controls, they have an intermediate amount. But if we look at all 4 DUP15Q subjects, they have double and sometimes triple the amount that the controls have. And you do not see this in paternal duplication, and you also do not see this in Angelman syndrome. So here's the group data. So this is we just averaged all controls, all the dupes, the paternal dupes and Angelman. You can see they're almost identical to each other, and the DUP15Q neurons are about triple the amount of action as the controls have. Now, there's a lot of things that could cause this, and I'd like to have you think about a car that moves too fast. So first off, the car could have a really powerful engine. That could be what causes it to go really fast. It could have some really advanced sort of tires that make it, reduce the friction on the road and make the car go faster. The body could be made up of very light material that could help make it go faster. Or it could have something that just doesn't have brakes, so it always just constantly accelerates, right? There's a lot of things that could change speed, just like there's a lot of things that could increase the action that we just saw in the DUP15Q neurons. So really what we did is we run through a bunch of tests for these different parts of the cell to see which could be causing this increased action. So you're looking at here again is blue is control, red is Angelman, and purple is DUP15Q. And this first sort of 8 or 9 dots is just baseline. So they're all sort of matched, you can see that. And this gray area is when we do the test to see which of these issues is causing the increased activity. And the take home here is that if you look at the purple versus blue, here, here, and here, there's not much difference. That means that when we tested these things, the results were the same between controls and DUP15Q, meaning that this is not contributing to the actual increased activity. However, if you look at this one issue, we see there's a large difference here, a whole gap between the blue and purple, and that includes differences between the error bars. So these are really quite significant differences. The interesting thing about this particular part, it's called KCNQ2, it's a channel. It actually results in something called benign familial neonatal convulsions. So these are babies who are born with seizures that then later get better, and then they never have a seizure again. But these are actual true mutations of KCNQ2. And the way this thing works is that it acts as a brake on activity. So you can actually press on the brake or block the brake, and then if you press on it, obviously you will reduce the speed or reduce the action, and if you release the brake or block the brake, you'll stop the dampening of activity and increase the action. So we tested this on our control neurons. This first bar is the actual baseline that I showed you. The second bar is when we press on the brake, and you can see it goes way down. And the last bar is when we block the brake, preventing the slowing down, and we can see it goes way up. If we do this in Angelman syndrome, first bar is baseline, second bar, as we're pressing on the brake, we're slowing them down, the bar gets smaller. The last bar, we block the brake, prevents the slowing down, gets much bigger. And if we do this to our DUP15Q neurons, this is again from 4 patients, we see that the baseline activity is much higher, which I already showed you, double, triple. And then when we press on the brake or block the brake, we don't get the same response we do in controls and Angelman neurons, meaning that the brake is disrupted. This particular channel that could cause this increased activity is actually not working the same as it is in controls or even Angelman syndrome. So that's the second sort of take-home point. The last take-home point I'll talk to you about is turning down the volume. So if we go back to the chatter, I showed you there's small events and there's large events, and they can get bigger and bigger and eventually lead to an action potential. It turns out that if you challenge neurons and you make things way too loud, that they like to turn it back down, right? Because being too loud constantly is obviously not going to make you very happy. So this is a scene of Neil Young's sounds. He's sort of famous for having these large amplifiers in the back. So too loud makes you a little grumpy, not happy. And then we'll turn down to a small Marshall amplifier. This is much more satisfying. So we do this now in our cells. So we give them a challenge to make them turn up the volume and see if they can turn it back down. Here's control in this first graph, Angelman in the second graph, and DUP15Q in this last graph. This blue line represents just baseline volumes. So these here on the x-axis is just size of the different volumes. And so you can see there's a range. And this green line here, this dotted line, shows that when you turn up the volume, the cells in control shift to the left, meaning it can reduce their amplitudes or reduce their volumes. So you turn it up, and all of a sudden they can shift to the left, making their volume smaller, which is what you'd expect. The Angelman are almost identical. I mean, you could think that I just took this graph here on the left and just copied it. So here's the Angelman. The blue is their normal volumes. When you turn it up, they become green, and they turn their volumes down. If you look at the DUP15Q graph, you don't see any difference between any of the dotted lines or the control. So there's no shift at all. They cannot turn the volume down when you've turned the volume up, as the controls in Angelman neurons do. So the take-home conclusions here are that neurons from DUP15Q patients are hyperexcitable, so they have lots of activity, sometimes 2 or 3-fold. This hyperexcitability is due to an impairment in a brake, sort of like a car brake, in terms of not dampening that activity. So this brake is sort of either broken or doesn't work quite as well, and so that increases this activity. And the last thing, and this just sort of adds on to these other problems, is the fact that it cannot turn the volume down. So the fact that they have lots of this activity, you could usually turn this activity down, but one of those mechanisms is impaired in neurons from DUP15Q patients. So the interesting thing about all this is that we have this target, this brake, KCNQ2, that we think might be a large role player in all these different features. And it's, again, related to other forms of seizures, and there are drugs out there that we're trying on our cells to see if they can respond or not respond or overcome this impairment. And so we're sort of doing that right now as a future objective in this next year of the fellowship. So with that, I would like to thank the Levine lab and the Chamberlain lab, who's the one who's gotten us these cells and is our collaborator, as well as the DUP15Q Alliance, who's supported this work, and all of you. Thank you.
Thanks for coming to hear me talk. I'm really excited to be here. As Guy mentioned, my name is Kevin Hope, and I'm from Dr. Reiter's lab. You may be familiar with him from the tooth studies, but we have another aspect in the lab where we actually use fruit flies to try to figure out what's going on with some of the genes that we're working on. So these are the same fruit flies that you might see if you leave some bananas out at your house and you get little tiny flies flying around. That's what we use in our lab. So your first question is probably, why would you use fruit flies to study humans? Well, humans and flies are actually remarkably similar. They share about 75% of genes that are believed to cause diseases. And the basic cellular functions are conserved between flies and humans. So just like cells in humans need to have energy, need to have inputs and outputs, need to be in a happy environment, cells in humans and flies need to do the same thing. And flies actually have quite complex behaviors. So they can be aggressive, they need to groom and clean themselves, they need to sleep, they need to mate, they need to eat, they need to do a lot of the same things that we need to do. And the human brain is actually really complex. There's about 86 billion neurons, so that's a lot of neurons to try to study at once, but flies only have about 135,000. So it's a little bit more reduced, like I was mentioning, but it can make it a little bit easier to try to figure out what's going on there. So as James mentioned, brains are kind of made up of this one popular cell type called neurons. So neurons are the electrical cells, and that's what a lot of neuroscientists focus on. But there's also another cell type in there, and those are called glia. So in the diagram over here, the neurons are the big yellow cell. They're what's firing the action potentials and have all that electrical activity, whereas the glia cells are these smaller cells that will support the neurons. So they will provide the neurons with energy, they'll keep the environment that the neurons live in happy, and they'll provide cues that neurons need to grow and develop. So one of the first experiments that I did was I wanted to look at how having too much of the protein UB3A, which I'm sure you're all a little bit familiar with, how having too much of that protein could be affecting different aspects of DUP15Q. And the one thing that I wanted to focus in on was seizures. So the first thing I did was I made flies that had too much UB3A in their neurons. and I tested them for seizure behavior. And these are actually flies inside of little tubes. This fly in the tube over here on the left is a control, and the fly in the tube on the right is a fly that has too much UB3 in its neurons, and I tested them for seizures, and I hope you can see that this fly is actually up and it's walking around just fine, just like its control fly. But when I put too much UB3A now in the glial cells, so those are those support cells, and test the flies for seizures, see this fly is laying here motionless on the bottom of the tube. And we'll just watch for just a second, but it's going to have some uncontrolled muscle activity and go through these very well stereotyped seizure progression in flies. So you see there how it's kind of moving around on the bottom of the tube? So that's actually a fly having a seizure. This has been well characterized going back into the 1970s. People have been using flies to investigate seizures. So this is a pretty good model, we think, for what's going on here. And so next I actually wanted to look into the fly brains, and believe it or not, flies have complex little brains. You can dissect them out and stain them with various things. Here is the fly's version of UB3A. I've stained that in red. And then here's another protein called ATP alpha, and it's actually a pump. So it's involved in keeping the external space inside the brain kind of happy, so not too much potassium or like kind of salts, keep salt balance. And you can see that the red protein, so that's UB3A, actually lines up very well with the protein I've labeled in green, which is ATP alpha. And this is a way to actually measure ATP alpha in the brains of flies. So here's control levels of that ATP alpha protein, and when I overexpress UB3A, we actually see less of the ATP alpha protein. So what we think is going on here is whenever the flies have too much UB3A in their glial cells, it's making the ATP alpha levels go down, and that's what's causing the seizures. So we think we're starting to get to a mechanism of too much UB3A leading to the seizure phenotype. And so that's great. We think we found out what's going on. But the bigger question is, what can we do to try to help with this? So the typical kind of drug development process, you start with maybe 10,000 or 100,000 compounds. You go through a lot of preclinical testing. You identify the handful that are starting to have the effect that you want. And the vast majority of these drugs are going to fail at some point. They might have toxic side effects, they might not get into the brain, they might do something totally different in a cell culture than they would in an actual organism, and then they might actually act different in people than they do in any of your animal models. But this whole process can take about 15 years from start to finish, so that's a long time. And this also costs a lot of money. So way back in 1950, if you invested $1 billion into drug research, you would get about 50 new drugs. And in 2010, if you invest $1 billion, you don't even get one drug. It takes about $2 billion now to get one new drug. So with this huge time hurdle and this huge cost hurdle, we're trying to think of ways that we can kind of get around this. So one way to do this is to take old drugs and try to use them for a new purpose. So James Black, who was a Nobel Prize winner, said, the most fruitful basis for the discovery of a new drug is to start with an old drug. So we kind of latched onto this idea. So we're going to start with drugs that are already approved by the FDA. So they'll already have safety profiles known, and that will save us some time and some money trying to get over those hurdles. And then we're going to use this drug repurposing strategy. And so this has been useful in a couple different disorders. There have been heart drugs that have been repurposed for neurodegenerative diseases, and there have been antibiotics that have then been used for anti-cancer, and I'm sure everybody's familiar with the story of Viagra. So we can cut out some of these steps going from drug discovery all the way to FDA approval, which takes 15 years and billions of dollars, to hopefully something that cuts down the time and the cost. So we're using a couple different drug libraries, one of which is called the Prestwick library. So that contains about 1,300 compounds, and all of them have already been approved by the FDA. And this library is actually picked because the safety profiles were very well known, and they were known to be active already in the brains of people. We're also going to try to go through the NIH clinical collection. So that contains about 700 compounds, and again, these have been selected for their safety profiles and their history of use in human clinical trials. So these might be drugs that have gotten to the end of a clinical trial and then failed, and they failed because they didn't meet the endpoint requirements, not because they weren't safe. We're really close to Ole Miss, and they have a natural product library, so they have about 500 compounds that they can give us. So these are products derived from like plants that chemists will just extract. And then we have another natural product set of about 420 compounds. So for this, what we do, well, I should also note that our lab received funding from the NIH actually to help fund this study. So we submitted this as an NIH grant and actually got it. So like I mentioned, the goal of this project is to try to find compounds that have already been approved by the FDA and then try to treat seizures in our flies. We're doing this in collaboration with a pharmacist at UTHSC, and we've already gotten through about 500 compounds. And basically what we do is we take our flies that are 2 different genotypes, and we cross them to get the flies that we want. So these are the flies that, like I showed you in the video, will have the seizures. And flies just live in these little tiny tubes with a little bit of food on the bottom. You can see some in the bottom of the picture here. And we just put the drugs in the food and let the flies grow up in those tubes, and then we test them for seizure behavior. And here's kind of our team working on this. So we recently got a postdoc to work on this, and then we have an undergraduate student who's helping us out, and then we have a technician. So all 4 of us are kind of trying to take this head on. And see if we can get through, hopefully, these 2,000 drugs that we have on hand, and there's plenty of other compound libraries out there that we could get to if we need to. So with that, I'd just like to thank my mentor, Dr. Ryder, the other people who I mentioned who are working on the project, and I would like to thank the DUP15Q Alliance and all of you guys for all your support. And I'm happy for any questions. We get through about, he asked how long it would take us to get through these compounds. We get through about 80 to 160 a week. So hopefully not too long, but that's just kind of the preliminary stuff. And then if we find any hits, we then do like a dose response curve. So, it won't take too long to get through them initially, but then there'll be some follow-up work at the end. Yeah, so he asked if there are any other like genetic mechanisms that could work through glial cells. Not really that I know of. The glial cell is the glial field's pretty new. That protein that I talked about called ATP alpha is actually known to be expressed in glial cells, and there are mutations in that protein that have led to like migraine and seizure in some people, so we think we might be on the right track with that, but the glial field's pretty new and there's not a lot of focus on it.
So I just want to say how investment, so we talk about fundraising, we've talked a lot about outreach and advocacy. We do fundraising, and we paid for the pilot study that they did at the University of Tennessee, and it came back 20-fold in an NIH grant now that is focusing on DUP15Q. So I just want to say, when we talk about fundraising, when we talk about our dollars at work, this is what happens. It triples, quadruples, 20 times. It really does pay. Every single dollar that comes in goes to these kind of research. Every time we fund a research project, this is the kind of research project we do. We do high-quality research projects. We target amazing researchers who are really going to make a difference in everybody's lives. Now you can hear them.
So we've had both our fellows. We've had flies. We've had stem cells. We also have mice coming soon. But before we do that, since we've just had both of our fellows, it's a great time actually to make a very exciting announcement. We have awarded another DUP15 fellowship for the next 2 years. And to make that announcement, is Carolyn here? Yeah. Carolyn Shannon, who needs no introduction, but he's a wonderful friend of DUP15Q.
Hi. Yeah, some of y'all know me from the olden days. Anyway, it's my pleasure to make this announcement. I'm part of the Professional Advisory Board, and this year and the last time I got to be the chair of the review committee for these applications. And I was really happy with the last two because you could see how great they've done. And we really thought that these model systems the last time would be translatable into something that we could use for treatment. So seizures in a dish would be something we could use to go on to try to find some way we could treat seizures, because as you all know, seizures are a major problem for some of our kids and for many of our kids. And the flies, again, we thought when we were reviewing that application, we said this is a way we could use them for drugs. And that the review committee said we could use these things to screen drugs, and look at here. So this time when we were reviewing applications, we said we're going to review them not only on scientific merit, but on things that we could use that were completely relevant to the kids right now. And so we had 8 applications. They were from, I guess, 5 institutions, and I was able to pull together a review panel of 12 people who were it was a great panel. They were experts in the areas. I managed to tap people who were complete experts from outside that knew the work that was being done. And in doing so, they were all very good, but one rose just to the top based on relevance. And that one was from Shefali Jesse's lab, which you all many know. And the winner is Vidya Saravaнаpandian, long name. I practiced saying it many times. And she's a second-year graduate student in the Center of Autism Research and Treatment program, or at the Center of Autism Research and Treatment institution at UCLA. She's actually in the neuroscience training program. And I used to be at UCLA, and that's a very elite graduate program. It's really hard to get into. And she's doing exceptionally well. I'm just going to tell you a little bit about her so you know what exactly the quality of the person that y'all just awarded an application for. In doing so, during the training, they have to rotate in different labs, and she picked labs that would put her in the optimal position to work on DUP15, which was Dan Geschwin's lab, who works on the genetics of DUP15, and then Payman Gulshani's lab, who works on some of the neurobiology in DUP15. Prior to coming to Shefali's lab, the project's name is Evaluation of a Scalable EEG Biomarker in DUP15 Syndrome. So that's what she's going to work on, her EEG work. And she did her undergraduate in Bangalore using a rat model of brain injury to examine ways to repair brains using transplantation systems. She then went on to do a master's degree in a rat model of malaria, which you know is a big issue in India. And again, to me, that just shows her commitment to looking at problems that are directly relevant to an issue that is pertinent to the population where she lives. Then she moved to the US, where she started working on autism. And she was at Stanford for 4 years working with Theo Palmer. And then at that lab, she did some work on brain development, including looking at that GABA beta 3 gene that we talked about yesterday in the genetics work. And that was looking at the role of GABA beta 3 deficiency during brain development in utero, so during gestation. So again, that puts her in, she's got like insight into so many pieces of DUP15 from just early on. And so I think, and that part of it is just amazing, because she just puts her in this position where she knows already so many things, and then now she's in this great training program. She's been working on understanding and learning how to do the modeling in the EEG, so that she's really in Shefali's lab already as a second-year student. She's been learning how to do the modeling in the EEG system, so that she already knows how that part works. As a graduate student, she's attained several awards, including being on the T32 grant from UCLA, which is a predoctoral fellowship. Again, that's another thing that they don't award to everybody. I was lucky to have a neuroscience training student when I was there, and I can tell you it was really hard to get a neuroscience student, and to get them on the T32 program was, again, something that you really fought for. She's gotten neuroscience outreach funds twice, and she was awarded the DUP15 Travel Award in 2016. She's also exceptionally well published for a graduate student at this stage of her career. She has 10 manuscripts and publications and presentations at major meetings. The work that she's going to do, looking at the scalable biomarkers, essentially look at those beta oscillations that they've identified in DUP15. And a biomarker is for those of you who don't really understand what that is, it's essentially a non-biased way to find something that will tell you whether or not something changes in the syndrome. And the EEG change that they see is this change in the waveform on the EEG. And what they want to use it for is to look at whether a treatment will change it. They also it will allow them, although it's not really part of the study, but again, looking at how things get used longitudinal, is if they follow kids over time, does it change over time? And how does it correlate with phenotype, which is part of their study? So our kids who are more severe, do they have a shift in how that curve looks? And in how the kids change over time, will we be able to say that curve shifts? If the kids have a change in their symptoms, does that waveform shift with them? And again, if we get treatment, can we see them change back to a more normal curve? Because we see that some kids have a bigger shift in that beta curve versus a smaller shift. So can we make it so that if we get drugs, can we shift it back? This is really important work. And as the people that are enrolled in this study are really contributing to our understanding of DUP15, and it will, I think, move the project forward. And as we get drug studies going, here she is. I didn't realize she was standing over there. Come here, yeah. Come here. I'm bragging on her, and I didn't see her standing there. Yeah. So everybody congratulate this girl. But anyway, so I should let you tell a little bit about what you're doing. But
Sure,
yeah.
absolutely. Well, thank you so much,
Did I describe it even remotely well?
No, I think you did it better.
Okay.
You did it better than what I would have explained, but yes,
Okay.
Thank you so much. First of all, hello everyone, and first of all, I would like to start by thanking the DUP15Q Alliance and everyone in the committee, and I think this is such an honor for me to receive this award. So I really appreciate that, and I'm really, really excited to be able to move forward with our ideas. I've been working in Shefali's lab for the past year, but this is my first ever family conference, and this has been such an amazing opportunity for me to meet with you all. And I see a lot of familiar faces. Like, I met with a lot of you, I think, in the past 2 days, and it's been so great to meet with all the kids. And I think I'm hoping to meet more of you today at the EEG sessions later. At this point, we've already collected data from like around 30 kids, which is amazing. So I'm a second-year graduate student in Shefali's lab, and I've been looking at EEG biomarkers. And I think you've already heard Shefali speak a few times yesterday, talking about EEG biomarkers. But importantly, what we really hope to move forward with this amazing grant opportunity is to really be able to scalably measure EEG and be able to have this available for everyone. So right now we are collecting EEG with like a sophisticated high density 129 channel electrodes. And what we see is we are able to do the same processing methods that we would do with this sophisticated method. And it is comparable to clinical EEG that we would collect at the different DUP15Q clinics. So our goal is to actually pull in data from across DUP15Q clinics across the country. So I'm really hopeful that that'll help us really understand the signature better, and also correlate, if we think that this EEG signature that we are seeing in individuals with DUP15Q relates to the underlying genetic variation, then if like having a lot more or a bigger, much larger population will give us a better understanding of how that relates to their cognitive abilities and developmental outcomes also. So I think in that sense, it's such an amazing opportunity. So when we move forward with the clinical trial, we would be able to have this available for everyone across the country, and we will have some meaningful understanding of the syndrome. And I think I'm also really, really excited about working in DUP15Q syndrome, because I've seen from my conversations with Shefali, like how much the community has grown ever since DUP15Q Alliance was started. I think that's to all you guys' hard work. So I'm really excited to be able to work with this community that is so strong and working so hard towards moving forward to have an better understanding of the syndrome. So thank you so much again, everyone. Thanks again for the work.
Are James and Kevin still in the room? Stand up. Once again, give it up for our DUP15 fellows. Doing all this great work for. There's only 3 of them. I wish we could afford many more, so please donate more money. And they're doing all the hard work, and remember, we're all counting on you. So to round out our sort of set of models of reality, models of DUP15Q, which we're using to help drug companies find drugs that are going to help you all, we have Jill Silverman. Remember years and years ago we funded those mice, those DUP15 mice? So they didn't just all go die in the corner. They before that, well, I mean, some of them are dead now. Sorry, but before they gave it up, they were well and truly analysed by Jill Silverman and her colleagues at Mind Institute to figure out, is there anything different about these DUP15 mice that drug companies can look to fix and see if it's correctable in the mice, then it's correctable in the humans. So it's the same game plan. Jill is rounding out the story, and I'm excited to present. I'm going to put your little thing in here.
Thanks, Mike. I'm so excited to be here to get to present to everyone today and to talk to the Alliance from the epidemic. Oh, okay. I'm going to talk up here when I talk the microphone. I'm just waiting for him. Okay, thank you. So I'm really excited to meet with you all today. I have to just add one personal touch, is that it really is because of Katie Luxinger and Guy that I'm standing here right now. A few years ago, the Duplication Alliance meeting was at the Mind Institute, and at that time I was basically studying a few different models of autism spectrum disorder, and it was Katie and Guy's tenacity and saying, you know, and texting late at night and saying, you know, this is why this group is really interesting, and we're going to get you all the human data that you need to really be translational, and this is where your lab could go. And now this is all my lab works on, so it's been amazing. Okay, so the audience does seem to know about the mouse models then and how you use different model projects, and so. I want to talk about what and why. So I know it's a hard concept to go from being a parent and being highly involved with developmental disabilities community to constantly reinforce the importance of animal models. And it's because our kids are human and we're human, and there is no way to just look in the brains and have the brains while they're alive and awake and behaving and having disruptive behaviors or having behaviors that can't have our kids follow all neurotypical standards. And so even though the condition is human, the animals can really provide insight into the underlying biology. I mean, essentially, we're more than 90% similar to a rodent.
And so our labs generally use animal model organisms to investigate the genetic causes of disease. And then once we find phenotypes, like they have learning and memory impairments or they have seizures, then we're able to use these same animal models and go back and test various drug treatments. So very translational. But it's also important, every student that comes through my lab will know very quickly that if they say autistic or DUP15 mouse or something, that they're not allowed to say that, and that these are model systems. The mice don't really have DUP15Q, and the cells don't really have DUP15Q. These are model systems, and so there's that. And then this is just a funny kind of picture that because I'm always saying models, 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 and then I was teaching this course, and the freshmen were like, what are animal models anyway? And I'm like, well, here's examples of like model pictures, but animal models for science are not model pictures. We use model systems in animals because we need them to behave and we need them to do complex behavior. Cells, for everything that we use them for excitability, because that is what the brain is, they don't behave. So we need a live, awake, behaving animal. And so they're used for every single type of biological disorder. I mean, everything that we know about cancer or how to take Tylenol when you have a fever has all been developed in animal model organisms. And so for the brain, this is really hard because we're not just talking about a disorder of the brain, which controls everything, but you're talking about a developmental disorder. So we need to look at brains at many different time points across development and make sure that if we see changes, that we can see them later on, that we can go in and rescue later on, and it doesn't just have to be early. And so that's why various animal models come into play and are important. And so after I said this meeting in Sacramento, I spoke a lot with Guy and Katie about the duplication group, and I got really interested in working with Scott Tindow, and Larry Ryder, who had been working on this new animal model. And this new animal model overexpressed this gene, UBE3A. And the reason that their animal model was even really spectacular was that not only did it overexpress UBE3A, which is one of the genes in the 15Q, and it's the gene that scientists that are really interested in because it causes Angelman syndrome. And so a lot of effort has been focused toward UBE3A. But from your own clinical data that all of you families have participated in, with Stormy and other groups, you've seen that there is elevated UBE3A in cells. And there's other cases of individuals that don't have a full duplication, but just overexpress UBE3A that also have developmental delay, learning disability, and an autism relevant profile. So this gives us really good evidence that UBE3A is key. And so the strategy that my lab uses in general for testing how this gene would work is to take these models up in A. There's a few different models of overexpression, but the one that we use is amazing because we can reverse it on and off and make sure that the gene is causing it and works, and I'll tell you about that in a minute. We do this comprehensive phenotyping. I spent a lot of time at the Mind Institute and with many clinicians, clinical psychologists I came from the National Institute of Health before I moved to the Mind Institute. I spent a lot of time, as many parents have, on the opposite side of that window that's one-sided when the assessments are being done, to make sure that the behaviors that we design in our lab are ethologically relevant because we're looking at a mouse or a rat, but at the same time relevant to what the kids are going to be tested in so that they have some crossover. And then we look at behaviors across development and we find phenotypes, and those phenotypes, including EEG, which you just heard a lot about, will be able to be used as markers for therapeutic intervention. And so since we're short on time, I'm gonna skip over the original two models of DUP15 because we never got a chance to work on them for various reasons. But the model that the Alliance originally funded is this model, which was just published in Human Molecular Genetics, literally on July 19th. So when I say just, I'm not joking. And I'm gonna share with you some of the data from that paper today. But the thing about this model is it's UBE3A specifically, so it's no other genes. And after we've identified many phenotypes, I'll tell you about them today, that are relevant and analogous to many of the symptoms that are in the children with the duplication. But also now we have the opportunity to reverse in this model system and prove that we have the right target. Okay. And that's a blot, which is really not as impressive as my behavioral videos, but it just shows that the model system is working. You want me to use this cursor here, right? It just shows that there's lots and lots of UBE3A in our model system. There's just the normal amount and the controls, and that the system, the chemical that we add that would go in and reverse it, does so quite nicely, as you can see by like the white bar in the bottom and that asterisk mark there. So we know the system works and we know that it reverses. And so let me tell you about some of the phenotypes that we found. Well, here's where I'm gonna have a problem because of my video audio conversion. So who wants to hear videos and audios, or who wants me to just keep going? What do you think? Okay. So when the pups are separated, so let me actually go to the UBE3A side. So here's our transgenic mice. This is basically your well check. You bring your children in. These are milestones that they get checked. We check how fast they do their writing reflex and how much they weigh and how long it takes for their eyes to open. And all of these various normal developmental milestones that occur in mice, we checked them in our mutant mice to see if there was delays in our transgenics. And the most interesting thing that we observed is this delay or less number of ultrasonic vocalizations. So you're right, mice, you're thinking mice don't talk, and they don't. But what they do is they vocalize in a frequency that is above the human hearing range. And so it's it's a little above 40, 50 kilohertz. And then they not only call, but they call in these really unique shapes. And so, although they're not going to play because this isn't my computer, they call in shapes they go upward, they go downward, they have multiple different steps, so you can see what they look like here. And we saw that these model mice of overexpression UBE3A had altered calls. And that's really cool. And anybody that's interested in hearing these pup calls afterwards, come see me, because all of these videos and audio files are on my Mac. This is task of elevated plus maze. The mouse is a mouse. It would prefer to be where, I mean, many humans are like this too, but would prefer to be where it's enclosed or safe and not out and exposed where the predator can get it. And so this apparatus over here is literally a cross, and the one side is open and bright and scary, and the other side is enclosed and more safe. And so traditionally in pharmaceutical companies, we think of this as a measure of anxiety, because if a mouse is just, walking right out on the open arm, they're not really concerned about their risky-taking behavior, whereas when they stay in the closed arm, they're being more cautious. And if you're to give an animal like a benzodiazepine or any typical anxiolytic, they go right out on the open arm. They're much less stressed out. And so what we saw when we performed this task on our controls, and then that complicated genetic nomenclature is our model mice, we saw that the model mice did a lot less of this open arm entry behavior, suggesting that they had a good deal of anxiety. And we saw that once, and we saw that twice, and then we saw it 3 times, but that's not on the slide here. And then we did 2 additional tasks that are very standard in scientific literature that also measure anxiety, and saw the same exact phenomenon. So this is 3 different tasks, many different replications showing anxiety. This is different types of vocalization that occur during play behavior. And so what you can see, but not here, and again, if you want to hear, just come catch me afterwards, is that there's a very high, distinct 50 kilohertz whistle sound that the animals make when they play. This sound means I'm happy. Woo, 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 woo. Like they whistle it when they're playing with one another. When you put them in the same area that they've played before, they start doing the whistle. They know that that's associated with positive reinforcement and social interaction. If you were to show a mouse a rat, or a rat a cat, because that's the way evolution goes, they will emit this call, which is slow and low, and at 22 kilohertz, they're saying, I'm afraid, don't come near me. These are the type of calls. So rodents, the point of showing you these is that there is communicative functions that we can measure using the rodent models, and that we plan on measuring in the DUP models. I'm gonna skip all of this about social for now, because there's a few videos that I really wanna go through to show you guys that are really cutting edge, sort of. So one part that the Alliance funded was to see if these model mice have deficits in their learning and memory, as we would hypothesize given the delays that we see in our clinical population. And this is a task that my lab and I have sort of optimized for developmental disabilities. It's been used more classically for aging. And so what it is it's an iPad. It's an iPad back here, and you'll see that in the video. And in this iPad system, the mouse gets some strawberry milkshake up here if it presses the right image. And so it's gonna go in slow motion now, because it's really fast so that you can see it. But in this task, it's asked, is the spider correct or the plane correct? And the spider's correct, and so he knows that, and he touches the spider with his nose, and then he comes back and gets strawberry milkshake reward. And so this animal is a great example of an animal that can learn the discrimination very easily. But the duplication animals, or the model mice, have trouble with the discrimination task. And what we're trying to do now is to do this simple version, which is 2 choices, and maybe make it so for the animals that have learning deficits to have even a simpler version where they're just looking at dots of light as opposed to complex images. But for the animals that do really well, to make it the task even more complex. Give them, make them know that spider is right over plane and that a daisy is correct over a ship's wheel. And so we're adding in different levels of discrimination to make it a little more challenging. And so we're hoping that the degree of learning deficits can correlate with the gene dosage. Okay, so where am I at in my talk here? Okay, playback, we're gonna skip that. You said I'm at 2 minutes, so here I was saying, so this last one, okay, so let's talk about this one. So there is this other really complex task of inference learning that we're working on. I don't have data to show you, but it's just like, oh, I don't know why I lost it. You all remember these logic problems, like Jane is taller than Sue, and Sue is taller than Jack, and Jack is taller than Alan, and you know that Jane is taller than Alan, even though I never said that, just because you inferred it. And this is really difficult for children with developmental disabilities. And so we've developed this task in the touchscreen. We show that the control animals can do it really well. They can always remember this. This is the human version on the left. We made similar analogous ones with our touchscreen images. And first, they do the anchor pair, which is really easy because the first one, like Jane has always been the tallest and the shortest has always been the shortest. But then we're actually also able to get them to make the discrimination between the middle pair, that the plane is always correct over the marble array. And so that task takes a really long time. I know I only have 2 minutes, but I really want to show you this because we just talked about EEG. So most importantly and most interestingly is that these mice that overexpress UBE3A, while we weren't able to, I'm not saying that they don't have spontaneous seizures because we are not in the colony 24 hours a day, and we did have some animals that were dying, but we couldn't get genotypes on them right away. So what we ended up doing to have some data on this was to give them a very, very low dose of a convulsant to see if the animals that had the mutation had faster seizures and were more susceptible to it. And we saw this over and over again, that the animals that overexpressed UBE3A had a really fast seizure. They were faster in everything. We used EEG to corroborate what we were seeing in behaviorally, and so now we're fairly positive that we have this EEG marker in the animals as well. And I think that Carolyn kind of already described why EEG is important. You all know from participating in Shefali's EEG study why EEG is important. But what I didn't know, if you knew, is that we can get these great EEGs in mice too. My student is here, my graduate student, Nicole Copping. She's right there, raise your hand, with blonde hair. And she has been implanting all of these duplication model mice with electrodes and collecting EEG data on them. And so we're hoping that we're gonna see what Shefali sees and then be able to simultaneously use that as a biomarker. This is what it looks like in a human with lots and lots of electrodes. We have a tiny little mouse brain. It's not that, we can't get that level of signal, but we do that. And with that, that's it. And I wanna end on a really optimistic note. So the Alliance had given me funding, seed funding, to start this project and get these mice and characterize them because they were made in Texas, and my lab is classical behavioral characterization lab. And so the Alliance funded that, and then just in April, we were awarded a $1.2 million NIH grant to further study this project. So that was all from you guys.